All right, so these are the vocab terms that we started class with. These are the things that you should know. We just watched the YouTube video that's linked in Canvas, so those of you watching our video at home, those are things you should do. Now we're going to go through and we are going to look at the section notes on mitosis. So when we look at mitosis, this is the actual division of the cell. So G1, S, G2, these are the interphase. 96-ish% of the cell's life is spent preparing for the division, doing its normal thing. Whatever that thing is, if it's muscle cells, it does muscle cell stuff. If it's nerve cells, it does nerve cell stuff. Now we're going to talk about the M phase. So of the cell's life, only about 4% of it is actually the mitosis piece. So mitosis is the division of the cytoplasm. It's the division of the nuclear information, the DNA, the chromosomes. And it's broken down into 4 steps. And to maybe help you, we have a puppy, okay? And our puppy's name is Mitosis. It's a cute puppy, but it's a puppy. It hasn't learned not to pee everywhere, all over the floor. So we're working on it with mitosis. But part of the process while we're here at school learning, she's at home and hopefully not peeing on the carpet. So what do we have? We have a pee mat for her to use just in case she can't make it till we get home. You know what I'm talking about? Those pet pee mats kind of thing, part of the training process, right? So why am I talking about a pee mat? Because that maybe is enough of a memory device to help you remember the 4 steps of mitosis in order that they go. PMAT, prophase, metaphase, anaphase, telophase. Sometimes telophase is pronounced telophase, depends on who you talk to, but I say telophase. So prophase, metaphase, anaphase, telophase. And then a lot of times when we talk about telophase, I often will talk about cytokinesis that kind of occurs as well. Telophase is the division of the DNA, the chromosomes, where cytokinesis is more the cytoplasm, the cell membrane. But they kind of occurring at the same time. So when we talk about PMAT, there's also telophase. At the end of telophase, there's also cytokinesis. So that was what that video talked about, plants versus animals, cell plate. We don't really get too much in the differences. So important terms, we've already talked about these. We've talked about chromatin being uncoiled DNA, like a bowl of spaghetti. I think this slide we've already used almost, we've for sure used the bowl of spaghetti one. But the idea is the more times we use these terms, the more familiar we're going to be with it. So chromatin versus chromosomes, sister chromatids, genetically identical to one another. Gene is the location, the centromere is what holds them together. We defined these terms last week. Homologous chromosomes, same. So these are homologous chromosomes to one another. These are homologous chromosomes to each other. We have 23 homologous chromosomes in humans because we have 46 total chromosomes. On the homologous chromosomes, we have the same genes, gene A, gene B, gene C, gene C. These homologous chromosomes have gene D. They're the same gene, but they may have different alleles for those genes. All terms we've talked about so far. So, getting into prophase. Prophase, PMAT, prophase. Big thing that happens is we get from chromatin to chromosomes, so from uncoiled to condensed, compressed chromosomes. The nucleus the nuclear membrane, which is the nucleus, the outside of the nucleus, starts to dissolve, and we start to get these little spindle fiber, these rope-like structures that are going to aid and pull things apart. So prophase, nucleus dissolves, chromatin becomes chromatid or chromosomes. We start to get these things called spindle fibers. PMAT, prophase, metaphase, meta, middle, meta, middle, meta, middle, middle, meta, meta, middle, middle, meta, meta, middle, M. So with metaphase, something goes to the middle. And we're all about the division of the genetic information and the division of the cell itself. But we're not ready to divide the cell yet, So if we aren't ready to divide the cell yet, what's going to go to the middle? The genetic information, a chromosome. Meta, middle, the chromosomes go to the middle. They talked about along the equator, I don't know, they said something about the equator. Okay? Ana apart, anaphase apart, PMAT, prophase, metaphase, anaphase apart, anaphase apart, anaphase apart, AA. What pulls apart? Well, the chromosomes pull apart. And when we pull chromosomes, the X-shaped thing apart, when we pull chromosomes apart, we get two separate sister chromatids. So the sister chromatids pull apart, ana apart, the sister chromatids pull apart. Telophase, cytokinesis, we kind of, again, group these together. But during telophase, what's going to happen is we start to reform the nucleus. The DNA starts to go from chromatid back to chromatin. So it's uncoiling. It's going back to the bowl of spaghetti. The nucleus starts to reform. And then the cytokinesis part is the pinching of the cytoplasm and the division into two genetically identical diploid daughter cells. We start with one diploid cell. We call it a mother cell. And when we're done, we end up with two genetically identical diploid daughter cells. They are genetically identical to one another. So if this was a skin cell, you now have two genetically identical skin cells. If this is a muscle cell, you have two genetically identical muscle cells. One diploid creates two genetically identical diploid cells. And that's part of the growth, repair, replace part of the cell division. The reproduction is going to be asexual reproduction, but this is the big things that happen and why it goes through that. Prophase, metaphase, anaphase, telophase, PMAT. Prophase, meta middle, ana apart, telophase cytokinesis. One diploid cells creates two genetically identical daughter cells. Okay? Just a reminder, maybe I'm the bearer of bad news, I'm not really sure, but Mr. Rather made sure to tell us to inform you that as of tomorrow we are not going to be doing soft release anymore, and there will be a bell at the end of the class that we will stay up right until the scheduled time. We're not leaving early. Okay? Too many disruptions in the hallways. So the teachers decided, and Mr. Rather listened, that this is the way we're going. So there are some times that the bell may ring and I might need those little bit of extra seconds, and that's okay. You get to stay for those couple extra seconds for me to wrap up those last few words. Okay? So we're going to let you out a little bit early today, but as of tomorrow and in all your classes, no more soft release. Have a wonderful day, everyone. We will see you tomorrow.